Hello, everyone, and welcome to our online lecture series for plant biology. Today, we're going to take a look at how those hormones that we talked about last time play a role in a plant's ability to detect its external environment. Today, we're going to try to answer the fundamental question of how does the environment shape plant growth? Because as you can imagine, there are a lot of external signals that can change the way a plant is growing. It may need to react to differences in where it can grow taller, where the sun is better available, where water is better available, or if it were to have the misfortune of tipping over at some point, as you may have seen in any of your forest hikes throughout your life, if it tips over, it still needs to figure out which direction the sun is and grow towards that. These various responses to the environment are collectively known as tropisms, of which there are two different varieties. Positive tropisms, where the plant will detect a signal and grow towards it, and negative tropisms, where the presence of a signal will cause a plant to grow away from where that signal is coming from. Now, tropisms were first discovered with a brilliant set of experiments that I'll summarize here. So we know that the growing tip of a plant is influenced by hormones like auxin. And the concentration at those growing tips is very high. Now, when we remove a portion of that tip and set light to be blocked from a certain angle, like with these blocks, so this is preventing light signal, we can twist the growth pattern of the stem with nothing other than a way to block the light. Taken to the full growth pattern of a plant, we can see that a tipped over plant, or even an upside down tomato plant as seen here, can respond to detect both gravity, the location of light, and ultimately optimize its growth habit in order to make best use of all the resources around it. This ability to respond to light is called phototropism. As shown in the experiment above, one of the most important hormones involved in phototropism is auxin, which, from last time, we'll remember is one of our hormones synthesized in young leaves as they're developing and trafficked to our shoot apical meristems. Both of our apical meristems, to the shoot and the root apical meristems. So what happens in that growing tip? Let's say, for example, that we're trying to diagram what was happening in the shoot apical meristem of this tipped over tomato plant in order to allow it to adjust to grow straight up towards the light. Well, at first, looking at the SAM, and we'll ignore all of the illustrations of leaf primordia here, we can see that the cells in the region of elongation on each side are identical. The cells in the region of cell division, right, in the L1, L2, L3 of your tunica corpus, well, they're not really changing their length, they're just changing their copy number. But if the stimulus was to come from a different angle, rather than originally, hold on, I guess I'll turn it, if the plant was originally something like this, growing up towards the light, and then it tipped over, the light is no longer arriving from this direction, it is instead arriving from this direction, still above the plant. What this will cause is a change in where oxen's being trafficked. As shown in the experiment above, it starts fairly concentrated at the middle. Remember, it is acropedally transferred to the tip and then basipedally transferred out afterwards. So to make this more clear with our arrows, it'll look like that to begin with. But with the relocation of where the light signal is coming from, the pin proteins will move. So that way, the transport is not even to the top and the bottom. It is instead going to be preferentially transporting down below because the light is being sensed from above. These pin proteins move because of the light sensing that occurs from photoreceptors. These photoreceptors are particularly paying attention to blue light signals. When the location where the blue light is coming from changes, the auxin gradient will change, causing cells in the shoot to elongate. And so what does this look like? Well, the shoot had that original horizontal growth, but then with higher auxin gradients here at the bottom edge, these cells will elongate much further than the cells above. Oops. And so what this will actually cause is a turn. By growing at different rates, you now cause one side to elongate more than the other, leading to a curvature in the growing tip. And again, this auxin is being shuttled to that lower edge by pin proteins, which are responding to signals from photoreceptors to blue light. The auxin is going to migrate to the shady side. Now, what does this look like in terms of live graphics? Well, we have a nice GIF to help illustrate this. In our shoot, you can see that the horizontal stem will eventually start to turn upward because of the difference in the size and the elongation of these cells on the outer or lower edge. Notice how much longer these cells here are compared to their counterparts on the other side. We see a very big difference between the length of this cell and the length of this cell. That is caused by a change in the auxin concentration. That is caused by a growth phenotype that is responding to where the blue light is coming from. And so in this way, a plant has phototropism in response to where the light is coming from and will cause changes in tissue development down the road. Now, this methodology where auxin causes elongation when it's at a higher concentration in the shoot will be different when we talk about gravitropism. In gravitropism, the plant is responding to the plant is responding to gravity, and the hormone playing a role is still auxin. But the receptors are different. The signaling molecules are different. Whereas above, in phototropism, we had blue light signaling to photoreceptors, and those photoreceptors moving where pin proteins were located. We will still have an auxin pin change, but it is not induced by light. It is instead induced by something called the starch statolith hypothesis. And this is because we're discussing gravitropism in the root. And what do roots not get a lot of? No light. So light will not be the signaling molecule, will not be the source of changing auxin gradients in the root, and therefore gravitropism, because they're not used, or they're not present underground. The root, which is also very sensitive to auxin, will instead have a different response to high concentrations of auxin. Whereas in phototropism, high auxin in the shoot led to elongation. We will see how in gravitropism, high auxin in the root will lead to the opposite effect, growth suppression. Because they're interacting, well, first off, there's a lot of genes that differ between the root and the shoot, but also the signal that's driving this, the perception of gravity, is done by those statoliths, done by something different. So gravity is perceived by statoliths. And statoliths are actually amyloplasts. So again, this is an artifact of the fact that we named them before realizing what they were in terms of organellarly what they were. But an amyloplast is a starch storage organelle rather. And because they are a starch storage organelle, floating in a liquid cell, they can sink. They can move towards the side of the cell that gravity is pulling them down towards. The statoliths respond to gravitational pulls. Now, when these statoliths sediment or sink to the downward side, they actually drive pin relocation. So what does that mean practically? Well, in this image from your textbook, we have our root cap and our statoliths and the indole acetic acid, auxin, being trafficked. We see that when the root is facing down, the basipetal transfer is equal on both sides, allowing the elongation zone to be even on each side. But when the root is tipped over, the statoliths, rather than being equally distributed across the middle of this columella, and the statoliths are typically found in the columella, they instead have preferential transfer to one side. And whereas we saw the side with higher auxin being stimulated in growth in the shoot, it is the exact opposite in the root. There's growth suppression as to where that auxin is highest. Pretty clever mechanism in both cases, dealing with the previous basipedal and acropedal transport. Hopefully now you understand why I needed y'all to understand those terms. And using that evenness of distribution to help direct you, right? Think about this as though you were paddling a rowboat. If you paddle equally on both sides, you will get growth in a straight line. But if you are paddling more on one side than the other, you will begin to turn that boat. In the same way that we have physics applied to paddling a canoe or a tiny dinghy, we have auxin transport running the same concept in helping a plant respond to either light or gravity. And we actually have images of these statoliths in the columella of the root. We see here at the 2 marker that there are some really dark, grainy structures at the bottom of these 2 cells. This is an electron micrograph, so a little bit different to interpret from your previous forms of microscopy, but it allowed us to capture where the statoliths were in a frozen tissue. This tissue had to be kept at negative 80 degrees Celsius to take this picture, so it's not like anything was moving. It was frozen in place. And we see that the statoliths were set in the direction of root growth. We also have done other experiments showing how root growth and directionality changes gene expression and therefore the presence of hormones like cytokinins. Remember, cytokinins are your hormones that are instigating cell division. Now, in cell division, or rather with cytokinins, we can actually have a dye that's a reporter for how much cytokinins are present in different areas. And when we take a look at a root that's either early on versus later in its response to gravitropism, we see that there's a shift in the expression of cytokinins when the plant is turned over. 30 minutes later, after growth was allowed, you see that there's still high expression in the direction where the root was turning, corresponding to our synthesis and transport of auxin. So it's pretty cool that we see a concerted effort by several different hormones leading the way as to where the root should turn. So again, we have a cool GIF showing how the root will respond. Again, we see transport that highlights what would be the equivalent of the shade side. But keep in mind, in the root, it's not the shade side. It is instead the side that has the statoliths sedimenting or moving down towards the bottom of the cells. So it's a totally different receptor, but it still results in auxin being at higher concentration in one side of the root, and that concentration change, concentration differential, causing growth suppression versus growth elongation. So then, if I show y'all these images from your textbook, hopefully you can help me interpret, based on statolith location, where down is. Because that'd be a fun game to play on a quiz, right? Based on location of statoliths, indicate where down is. Well, since the statoliths are located at the bottom of the cell, we can assume that down is directly below in panel A. In panel B, we see that there is a large portion on the right side, but also some on the bottom, meaning that down is likely in a downright direction, perhaps even more in a right direction than down. To the same extent, you can even tell a slight tilt in the growth of the plant because the statoliths are not evenly distributed on the bottom here. This side is empty, likely indicating that the downward direction is slightly to the bottom right in panel C. So in this way, you can use hints based on where things are growing in the cell, where things are present in the cell to predict root growth and hormonal transfers. Now, there are a large number of other tropisms that we won't really discuss in this class. I don't want to overwhelm y'all with the different receptors. I will point out, though, that I do expect you to know both, or rather all things associated with the gravitational tropism, the fact that auxin's the hormone signaling. Auxin is reshuttled to where it needs to go based on pin proteins, which receive their signals from the starch statoliths. And in our phototropism, the fact that auxin is again doing the signaling, pin proteins relocate where they're going, but instead pin proteins are receiving their signals from photoreceptors. Also, knowing that there's a different response in terms of which tissue you're going to see elongation in versus which tissue you're going to see suppression in. And that has more to do with the other suite of genes that are being expressed in the shoot and the root. But we also have hydrotropism, which you can imagine being very useful for a plant. Its ability to grow to water. It is a positive tropism in most cases. We have thigmotropism, meaning that a plant will grow away from touch. So if it, and maybe you've heard this from your grandma when you were growing plants in the garden when younger, stop touching the plants, you're going to stunt their growth. If they feel a lot of touching or pressure coming from the direction of growing upward, they will instead try to maximize some other apical meristems, perhaps growing bushier and reducing your overall yield. Now, of course, thigmotropism is incredibly important in climbing plants. Any kind of vine, like this cucumber here. The thigmotropism that this vine uses is detecting where it can tie its tendrils around, and you'll notice the tendrils themselves are just spirals, right? How do we create a spiral? Well, we have cell elongation that's greater on the outside than on the inside. This causes the tendril, which is a modified leaf, to start curling around its subject. Pretty cool, right? We also have heliotropism, which can be somewhat different than phototropism, which is the idea of solar tracking here. This is best observed in some wildflowers that tend to move their heads in the direction of the sun at the day, so they can track the sun's movement throughout the day. And then we also have thigmotaxis. Rather than changing your growth patterns based on where touch is coming from, plants can additionally respond immediately to touch. And no plant illustrates this better than one of my personal favorites, Mimosa pudica. So in this GIF, we can see that when the human touches the tip of the plant, the leaves will begin to fold in on themselves, thinking that they need to be less appealing to predators who may be eating them, or hide and reduce tension due to rain. So there's a couple different ideas here, and mechanistically, this is actually happening through rapid wilting. But Mimosa pudica is an example to the fact that plants can indeed move in the short term. Operates much more differently than muscle movement in animals, but is probably one of all of my plant biology students' favorite examples of plant movement. To kind of wrap up this section on tropisms, we also want to respond to a couple, or include a couple of different vocabulary terms, including etiolation. Etiolation is, again, if you remember from our photosynthesis lectures, etioplasts, or plastids, organelles, plant organelles, that stimulate the rapid elongation of the stem in order to get the plant to find sunlight. So here on the left, we have a plant that was grown without sunlight and a plant that was grown with sunlight. The plant without light thinks that it is so deep under the ground that it is just extending its stem on each side, trying to get out from under the dirt. The stem is etiolated. To the same extent, you can see etiolation in these seedlings as they were grown in the dark, and then a light was placed at an angle. So their etiolation helps turn them towards the light in that direction. We additionally have a couple of terms associated with seed dormancy. Dormancy being the fact that a seed will wait for the appropriate time in order to begin to grow. You don't want seeds to break dormancy in the middle of winter. You don't need them, especially here in the state of Michigan. You don't need them trying to struggle against the snow to push it up. They're not going to have any water or adequate amount of sunlight to grow. So seeds may wait for a signal of vernalization. Vernalization is associated with a full winter-spring cycle. What this means is that the seed will need to be exposed to a cold signal and then a spring melt in order to begin to germinate and break dormancy. We additionally have stratification, seed stratification, where simply a cold spell is enough in order to break dormancy. Once dormancy is broken through experiencing a cold spell, you then will have the seed wait for potentially a first rain. So in regions where you don't have snowmelt, stratification fills the same niche as vernalization. And me having grown up in Southern California, there were seeds that required stratification, but they really weren't ever going to get a true vernalization. We also have seeds that require scarification or some sort of mechanical damage. Now, why would a seed want to experience mechanical damage before germinating? Well, because that mechanical damage may be associated with something like passing through a bird's digestive system. When a seed passes through the digestive system, it can get ripped up. The outside is damaged, but the inside is completely still intact. I'm sure we've all experienced this in our own lives. Those seeds will then have that outer layer ripped off, enabling them to then perceive signals like water in the environment to start growing when water is available. All of this seed dormancy ties into the idea of a seed bank. The fact that plants will generate lots and lots and lots of seeds, but not all of them will germinate at the same time. Some of them are stashed for different years because they are not perceiving the correct amount or a sufficient amount of cold or a sufficient amount of water in order to break their dormancy. This allows plants to stagger their growth. So that way, all the seeds don't germinate during one year that happens to be a bad year. They instead have some seeds banked for the future when that bad year may be skipped over. And in these ways, the environment, in terms of longer cycles around the year for dormancy, gravitropism, and phototropism, can impact our plant growth. Our next impact on plant growth has instead to do with its daily cycle. This daily cycle, whether it's in a plant or in a human, is called our circadian rhythm. It basically operates on the fact that we have a 24-hour cycle. I've included this image of a typical human circadian rhythm. We can see that certain things like our sharpest rise in blood pressure, when testosterone is secreted, highest alertness, when your coordination and reaction time are the greatest in the afternoon, and then when melatonin secretion will begin, are all actually programmed based on day length. Now, of course, we have a variation of these processes across humans. There are many different chronotypes, so you don't need to feel like you are left out of this if this does not match you exactly. This is one generalized circadian rhythm. But we do have a response to our 24-hour day length because we live on a planet that rotates every 24 hours. So that way each side of our planet gets maximum sun exposure for a short bit of time. Now, this circadian rhythm is obviously controlled genetically. The three parts of our genetic control of our circadian rhythm are as follows. There is a set of genes called the oscillator. And oscillators help you establish a rhythm. An input set of genes. These help synchronize you with the time of day of wherever you are. Think of what happens when you travel long distance, like to another continent. Your 24 hours are now shifted because of your new location. And then you have an output set of genes. These cause your physiology to change. They regulate your physiology, meaning that they cause these shifts in the human experience when you need to shift them based on where you are currently. And so you think about perhaps the last time that you changed time zones quite drastically. Prior to my job here at West Shore, I was a researcher in Germany. Traveling between Germany and the States, particularly California, was a 9-hour time difference, meaning that if I got up in California and traveled to Germany and was teleported immediately, it would be the morning for me, but the evening for them. My circadian rhythm would not have the right synchronization. I would have to wait for my input genes to perceive the time change over the course of a few days and readjust my oscillators and regulate my physiology through the output set of genes in order to get me to sync up with my new German location. In plants, the things that are perceiving our day length changes are red light phytochromes and blue light cryptochromes. And the reason why these two matter so much is because the proportion of red to blue light coming from the sun changes seasonally. And so plants are able to regulate what they're doing based on the time of year because of the amount of light that's blue versus red that they're perceiving in their leaves. This leads to plants that are classified as long day or short day plants. So what's happening here is that every single day you have a conversion between your phytochromes that are detecting either red light or far red light. Now, a plant's far red light detectors are converted into red detectors during the nighttime, whereas a plant's red detectors are converted into far red detectors during the day. If days are really long, that means that the plant has a lot more far red than red, and meaning that the days are long, and that these plants will flower when they detect this overabundance or skewed ratio of PFRs versus PRs. Short day plants are the exact opposite. They detect when days are getting shorter because they accumulate too much PRs because they're not being converted back from being PFRs, and instead they will flower in short days. And so in this way, it is the amount of night or the amount of darkness that is actually signaling to plants whether they need to flower or not. And long day plants flower when days are long, and short day plants flower when days are short. Some great examples of our long day plants are things like oats, and these are obligate long day, meaning that they will only flower when days are long. You also have some facultative long day plants, meaning that they can be a little bit flexible, but you'll get barley and wheat. You can tell I have a preference for bread products, but also pea plants are long day plants. And then we have a slew of short day plants that you may be familiar with. Short day plants like cotton that need less, a higher amount of PR than PFR because days are shorter than nights. Cotton, rice, and soybeans, as well as cannabis. All those plants will flower when their PR levels are higher than their PFRs. What we've done in a lot of our other agricultural plants, or very commonly in our agricultural plants, is create or select for a lot of day neutral plants. Day neutral plants are going to respond to other physiological requirements, not day length. Like, am I developmentally ready? Have I grown enough leaves? For example, certain corn plants will begin to flower immediately after they've made their 11th leaf, or something of the sort. And do they have enough sugar in their stem? Contrast these with what we see within our day length sensitive plants. Short day plants, meaning that they will flower under an 8-hour regime rather than a 16-hour regime. And then interestingly, what happens when you interrupt the dark with a little bit of light in between that 8-hour segment? Some short day plants like cocklebur will not flower, despite the fact that they're getting 16 hours of night. They're getting an interruption. This is why it can be very important for you to not turn on the lights in the greenhouse during certain times when we're trying to get different plants to flower. A long day plant, in contrast, like henbane, will not flower when days are short, but will instead flower even if they only get 8 hours of sunlight because the night is interrupted by the sun. So I think that does a good job wrapping up all of our discussion of external stimuli that can help plant growth and development, whether it relates to flowering and circadian rhythms, or hormone trafficking and changing the directionality of growth. These external factors like tropisms and circadian rhythms are essential to understand whether you're trying to be a gardener or breed a new variety. Plus, they're very clever and very cool, right?